the hope would be potentially in my group to learn about these ideas of gene regulation and development in the context of seeing them as a biological computation. So in the complex of seeing the processing that the network does to a particular set of stimuli as a computation. And one can, of course, do this both on the microscopic or molecular level, where what you see here is a video from Mustafa Mir's lab. The green thing is a crucial transcription factors inside nuclei in the fly embryo. And you see that basically based on these stimuli, this particular transcription factor is then expressed as a function of time also. Or, and I think this is a sort of a cute idea that was proposed by the plant community, Yasmin Merose and other people, that basically you can even think of these plants as like performing computation to an external stimulus. So what you saw in this video was a light source on the left, and then these plants sort of grow towards the light source, so they clearly seem to sort of integrate the stimulus somehow, and then as soon as the light source switches, they grow essentially away from it. So the question is, can we learn something about these networks by thinking that these things are a computation? And to just also maybe clarify that thought a little bit more, I would say what makes biological computations or biological systems as performing computations interesting compared to maybe on the one side an engineering lookup table, and on the other hand, like a human decision, I would say that in the engineering lookup table, you've essentially at some point learned an almost deterministic representation of your stimulus. And the way you learn this is probably by using a huge ton of different variables. Whereas on the human decisions, you probably also have a huge ton of different variables, but you, I would say, have a much more probabilistic representation of the stimulus, and there are all sorts of values and Bayesian sort of decision making coming in. So I think the biological computation is sort of nicely positioned, hopefully in between, where you have maybe like, you know, still a large number of parameters, but probably not an infinite. You also have a sort of probabilistic expression of this map between stimulus and response, but it's probably not entirely deterministic as in the lookup table, not entirely noisy as in the human decision. So I think it's actually sort of a nice in-between thing that we can hopefully understand. And so the question, I guess, I would say that we can ask is basically, can we identify principles on the physics side of the narrative that allow for precise responses if we have these stimuli? And on the biology side, I would also say that it's interesting to actually maybe really distinguish between the different systems to say, you know, are there specific behaviors or specific constraints in a specific biological systems that maybe modify these principles somehow? And I think sort of in what we've heard in earlier days of the talk, like with sort of Kabir's ideas about optimization, and then also maybe in Jakob's talk about sort of, you know, is it the growth rate that's optimized or some lag phase? I do think, you know, that with these principles, I guess one of the questions could be, what is potentially being optimized? Like, can we classify what is being optimized potentially in different systems? Or maybe if it's nothing, that's still fine, but I still think that there is a nice approach to these optimizations. So essentially in that view, what I would like to talk about in this talk is to basically seeing this idea of information optimization as one principle, maybe that has shortcoming, but that maybe we can use to learn something. And in particular, because we're thinking about cellular computations in this talk, I would say that we have some sort of signals, which in all of my cases are some transcription factors or I guess Wnt ligands, so like developmental signals that are processed by cells or nuclei in order to give gene regulatory responses. And on the output side, we're typically thinking like what Arjishman has done in the level of cell fate. So I'm not going to here sort of sit down and write down the individual gene expressions, but I'm just going to think about cell fate. And then the talk sort of also has two parts. In the first part, I would like to talk about fly development. And in the second part, I would like to talk about sort of cell differentiation in these signaling pathways about Wnt. And just to give you a heads up of what's going to come, oh yeah, sorry. So in the first part, what we're doing, similarly to my introduction, was to basically optimize the map between signal and response, or rather optimize this compression. Whereas in the second part, we will optimize the signal statistics. So what is being optimized will vary between the parts. And in the first part, to give you a small heads up, what I'm hopefully will try to show you is basically that we will look at what happens at these binding sites. So we will look at information processing in the cell, in the nucleus, of transcription factor at binding sites, regulatory binding sites that can be enhancers and promoters that give rise to the gene expression that is important for cell fate development. And in that context, I'm hoping to show you that clustering of these transcription factors at the binding site can be beneficial, but that it's actually important what you look at in terms of what is it beneficial for. So what variable are you actually optimizing? And then I will sort of show that we can look at basically finding these regulatory regions with the information bottleneck, and that clustering can actually help us achieve these bounds. That part is a little bit sort of boldly phrased, because I will tell you that there are certain shortcomings of the models that I have not addressed, but I think in principle it's a first step in that direction. Okay, and I also wanted to highlight the people who did the work. So Tamara was a master's student with me, and it's a collaboration with also her current advisor, Jos Svanniken, and Eme is a former group member in my group. And yeah, so the concrete question, and essentially this entire first part of this talk is, can we understand the enhancer or regulatory architecture using these InfoMax approaches? So we will have a bunch of signals. The things, these compressed states that we will be optimizing or that we will think about are the enhancers and promoters, and specifically we think about literally as we think of them as literally stochastic regions where you can stochastically bind and unbind different molecules. Okay. And we do this on the example of the fly embryo, basically because the fly embryo has to some extent been established as a system where the information flow is to some extent important. That's partially just intrinsic to the fact that, unlike for mammalian development, when the egg is laid, the egg is essentially living its independent life. So you could say, there's a lot of pressure to not put too many molecules in this egg from the side of the mother, and to also not make too many molecules because you only have like a tiny embryonal egg available. So what's happening in fly embryo development is that you have these maternal signals. There's the famous transcription factor bicoid that the mother deposits at the head of the embryo. It then diffuses and is zygotically expressed, and then leads to these gradients along the embryonal axis from head to tail. There are crucial genes that are expressed in between, and then after sort of approximately 2 to 3 hours of development, you get still pre-blasted information, an embryonal egg that has already genes that are expressed in these stripes. And these stripes are crucially important for body part segmentations, like similarly to probably how you used to bees that have proper stripes. These stripes will later on give rise to different segments like abdominal segment 1, abdominal segment 2, and so on. And so we can approximate the cell fates on this level essentially by the positional information along the x-axis. The thing that's happening in between is that a few gap genes are being expressed after approximately also 1.5 to 2 hours of development. So the information flow goes essentially maternal gap body part segmentations. And on both of these levels, even though there are more, I'm of course presenting a slightly simplified pictures here, on both of these levels, there are enhancers and promoters that sense the maternals and express the gap genes, and enhancers and promoters that sense the gap genes and give rise to the segmentation genes and Hox genes that later on provide this positional information. I also wanted to, yes. So I think they are meant to be like first order, the main ones. There are also, so basically these segmentation genes, there are 7. The famous one is Eve, and I think there you can really check what regulatory regions regulate Eve, and it's really a lot of gap genes. It also admittedly has still a lot of, or like not a lot, but some bicoid. So the idea is that actually maybe bicoid opens these regions or regulates them somewhat, but I think first order, you can really say that it's the gap genes that regulate at least Eve. There is in super late parts of this last stage before you make this image, essentially, there's some thinking that the segmentation genes essentially have some sort of repressive regulation going on. And in addition, on this level, there are also what's called the Hox genes. And they are regulated, I think, partially by some of the gap genes, but also by others. So I would say first order, but yeah. So Eric would love that question. So I think Eric was always super fascinated. He was always like, why make additional signals? What people say, and I think Eric Bischoff, sorry, and I think what people say, and I think some people are less excited about this, is that the gap gene essentially provides some pre-segmentation to the segmentation genes. But then Eric says, like, why would I have graded? I still have hunchback here graded, and surely that's better than just having some sort of modular thing. So I would say maybe it's not clear, but the gap genes also, hunchback especially, which is the one that comes up the first, also has other functions, also regulates the Hox genes, also regulates other genes. So I think maybe you needed more genes than just the maternals, but I don't know exactly why. Yeah. Oh, you can suggest. Mm-hmm. That's actually a great idea. I think, yeah, that's very nice. I think there's some, because it does diffuse, but I don't actually know like how crazy the diffusion is in the flying beer, but that's actually a great suggestion. Yeah, okay, thank you. Okay, yes, and so we're looking at essentially the information flow. In this talk, I'll mostly talk about this part, but I wanted to say that the reason why I was sort of interested in this information flow is that in the past we'd looked at this part, and I just wanted to say that basically we try to essentially infer the enhancer and promoter architecture that read the gap genes and make the parallel or the segmentation genes. And what's sort of interesting is that regulation in biology, often people seem to be thinking this can't be an infomax, like useful target, because the enhancers that sense these segmentation genes are essentially a mess. So some enhancers make one stripe of one of these genes, Eve, others make two stripes, others are so-called shadow enhancers, so they just support each other. So how can we understand, in quotation marks, anything using infomax approaches? And I think of course, we can't understand it all. And of course, all we can do with these infomax approaches is to maybe to some extent give some heuristics for our understanding. But at least in work that maybe if I have time, I'll mention more precisely, we were able to sort of use simple models for enhancers and also more complicated optimization approaches. And we said, the fact that we need multiple enhancers and the fact that these enhancers need to respond to combinations of these gap genes, so not just to a single gap genes, that is actually optimal. So I'm just saying this here because basically I'd used a particular Infomax framework, the information bottleneck, to explain this. And so given that intuition, of course, then when there were certain ideas in the Princeton and in other communities that maybe information is not maximized because we can observe certain biological features that seem to suggest it's otherwise, I sort of wanted to understand that a little bit better. And the concrete thing that I wanted to understand a little bit better is this thing that Krishna had already introduced on the first day, this idea of, what happens when transcription factors cluster. And I'm using the word clustering here because, there's like this huge debate in the transcription community of whether, clustering just means that there's essentially some pleasant affinity of these two transcription factors, or whether it means that you really have some large liquid phase separated droplet. And I would say in the fly embryo, the consensus seems to be that at least bicoid won't form a huge liquid phase separated droplet, partially because the number of molecules that you have available are sort of order 10,000 at the front and like really order tens or nothing at the back of the embryo. So how large of a liquid phase separated droplet can you form? However, when you look at bicoid, you can see that it's highly heterogeneously distributed in the nucleus. And this is partially because some people suggest that other pioneering transcription factors that are expressed at larger numbers, that they clusters and the bicoid likes hanging around them. Either way, somehow there seems to be some clustering through some mechanism that maybe one doesn't fully understand. And the question was, how does that, if we really clearly observe this, how does that affect this information max idea? And partially, the reason why there was pushback, especially from people around Eric Rieschaus, against this idea of, or not pushback, but I mean in the sense that they were worried about this clustering, because there had been these old ideas that clustering increases noise. And they come from a variety of different sources, some of them in the language of the early Burke-Vassell bounds, where you basically try to calculate the noise bounds on sensing. So you assume that a particular binding site or a region of receptor senses a particular signal, and now you calculate the signal to noise ratio for this sensing. And then you get that basically as soon as you have any type of cooperativity in the binding, you get essentially a second term here. So that means that the noise in the inferred signal actually goes up. So that's sort of bad news. Also, there's work by Ned Windgreen that essentially had looked at clustering, in this case more in an Ising model, which we will do also in this talk, clustering transcription factors, that also says that this noise increases with with clustering and general simulations by 10 Baldus groups. So all these ideas seem to suggest that clustering or cooperative binding, but especially clustering, is actually really bad for this information flow. And so I guess to some extent, I was sort of nervous about this, because if we do so much work to do info-max approaches and then we see something in biology that's clearly not an info-max approach, like essentially, are we making a pretty obvious mistake? And so yes. Independent signals. Okay. Yes, so this is actually on the level of Bitcoin. So this is maybe simpler than just like adding additional transcription factors. So we're not looking at gap gene, like not this like multi concentration condensates that Krishna was talking on. So we're saying, like, let's assume I have one signal transcription factor, and now I want to read out the signal, but now this transcription factor is actually clustering. So you can maybe intuitively say, like, in the worst case scenario, if you have a gradient, let's say even like if it's a linear gradient, if you now have clustering, you will have clusters and no clusters, and that sort of kills to some extent your signal. So it's in that direction. Yes. Okay, great. Yes, okay, so what we'll do here is like a very, very simple, and I would say almost low dimensional actually calculation. The only thing where you have some high dimensions is sort of the state space of the binding. But basically, we are looking at Bitcoin as an input simple transcription factors that clusters. We are then looking at a binding site region, a single binding site region that's somewhat plausible because Hunchback also is expressed like that, where these molecules now cluster. So basically, clustering means that if a molecule binds, this other molecule has a higher probability of binding. And then we're looking at how this particular region essentially can give rise to cell-fate states on the output level. So we are introducing this variable C for this compressed state, for the compression that happens at this binding site region. We are looking at signals S, and we are asking essentially, I'm defining that ICS is the information that this binding site region can have about the signal. ICX is the information that this binding site region has on the output. And now we're essentially asking how well can this binding site region sense, have information about the output if these transcription factors cluster. To do that, what we need to do is that we need to make models for these binding site regions, because once we have a model for this binding site region, we can write down this conditional probability distribution PC given S, and then we can essentially check. So that's what I'm going to do on the next two slides, essentially. So what we've done is we've essentially claimed that downstream gene expression is a function of time averaged occupation. We've also done other things. So time averaged occupation of how many molecules are at this binding site. That then gives us a signal distribution PC given S and allows us to calculate both mutual informations. And we've used two models. The first is Hill function-based models, just because a lot of people in gene regulation use them, where you have essentially three free parameters, which is one parameter that determines the binding affinity of, now you say basically all molecules, limit of infinite clustering, all molecules bind at the same time. You have some parameters K that determine the binding, some parameters H that determine to some extent the cooperativity. And you always, because we're looking at time averaged occupancy, we always have another parameter T, which essentially says how much time do I actually have to express downstream genes based on my transcription factor signal, or how much time do I have to measure? Yes? The cooperativity in binding. So we are saying basically once they're bound, we are saying proportional to how much is bound, they are making, approximately they are making that many genes. So we are saying the thing we care about is the occupation. Yes? Yeah. No, sorry. So there is a map from C to output, which I actually haven't. So that's similarly to how in the information bottleneck I calculate that. So once I get C, I basically compare C to S and then calculate an output, which is the positional, and which basically I say, how much information did S have about X versus now how much information does my clustered signal have about X. So I also have a map from C to X, and X is just, in this case, the number of cell fates that I could possibly have along this embryonal axis. Yeah. Yeah, thank you. Yes. Yes, so this was the thing that I might have it on a slide somewhere, but basically I do, I would do this essentially, which is basically if I have PC given S, I compare to PS given X and integrate over all signals. Thank you. Okay. And then we have sort of, because these hill functions make me extremely nervous. I don't know if you have the same, but because we wanted to also make sure that we really do the proper thing. And so we calculate essentially in like an almost 1D Ising model, how do individual molecules bind and unbind, and then we get like a large transition matrix of states and so on. In this case, we have also 3 parameters, which is the binding energy, the clustering of these neighboring molecules, and again, the T, how long do you have for measuring. Then with both of these models, essentially, we try to calculate probability distributions, either through simulations or through, assuming that the probability distributions is Gaussian, that I have a particular mean, which corresponds to my Hill function, and a particular noise, which I can calculate, in this case, not through Berg-Pascal, but just through assuming that these binding, unbinding events are binomial. And in the second part, we essentially simulated it. We also tried the Gaussian part. And you can see that you can calculate essentially as a function of Bicoid signal concentration. You calculate the occupation of this binding site. It has a certain noise. It has a certain mean. And now we try to ask how much information do these guys carry. And so what I wanted to say is basically that it really depends on, to some extent, the measurement time. So if you have a short amount of time available to measure, it always seems that weak clustering, so a weak J or some finite Hill coefficient, increases the information. And the reason for why that's true is actually sort of, even though the signal, even though the noise would always increase as a function of clustering, but of course what happens, maybe similarly to what we had before, if you have high noise in this map between S to C, it's much better to just do a switch, because then at least you can differentiate 2 states well, as opposed to smearing everything out. And so that's why for a short, so basically when this measurement T is short, you will have high noise bars here, because you only have essentially one measurement, so only one shot to essentially guess your concentration. So you need this finite clustering. When you do a long measurement time, and you look at, and this was the mutual information between C and S. So this is the information about the signal. When you look at these long measurement times, so the Hill function still says, okay, finite clustering is good. But then for the Ising, so the actual stochastic model, we find that the information really decreases as a function of J. So that is the thing that Ned and other people had found, that basically when I have long time, it's definitely not good to have sort of clustering, because my noise is essentially so low that it's better to have a linear signal. But now the important point that I wanted to make is that it really does actually matter what you look at, because if you look at I, C, and X, you find, for the short measurement time, you find the same. For the long measurement time, you find that the decay is essentially much slower. So you find that basically for, I guess, very finite clustering strength, you're essentially still not losing information about the variable that you care about. And so I would say that really sort of shows that maybe a lot of this work, which is sort of, how well can we sense a signal, is really important. But it maybe, if you have an intuition of what it is that you care about, maybe that's the thing that you should try and calculate. Okay. And so now that we've done this, now that we'd essentially, said, okay, this is okay, but I should say this is an overstatement, because we have not in anywhere calculated or included the noise at the promoters. And the reason for why I think that's okay is because there are so many crazy measurement estimates out there for what this noise at the promoter should be. Then you would also have to go from RNA to protein. So I think the fact that this works is definitely a limit of the types of things that I've put in the model, but I wouldn't have known how to do better, I think, at the moment, maybe, yeah.
Question about the biophysics.
Yeah.
I guess there's these enhancers which are pretty far away.
Yes.
Earth,
yes.
And these constituent factors bind and they kind of come together.
Yeah.
And then they recruit the
yes.
polymerases.
Yes.
So, which part of this is being null here? Like,
yes.
Just assuming
super simple. So just the first part. So literally just the binding. And then I'm assuming that I have, so in this case, I only have even one enhancer. I have one enhancer that has multiple transcription factors bind to it, and that leads to promotion. So I'm not looking at what happens if I have non-equilibrium processes going on here, like mediator that could also make it better, or how do I think about multiple enhancer elements that maybe in some sort of 3-dimensional architecture of the genome come together and regulate the promoter. Right. So I'm doing the trivial thing, because basically the early results had sort of suggested that already in this trivial thing, I get that clustering is bad. And so I wanted to check how that's true. Yeah.
Thank you.
Or we wanted, yeah. Because I mean, you could of course be right and say, well, you know, if I have this type of architecture, maybe then I have some sort of dominating clustering effect where that is better. But I guess to some extent, the worry in these like small concentration limits would have been, so we actually wanted to do multiple enhancers. The problem was that, first of all, it's harder to simulate, but to some extent, then you have to really think about, you know, what is my transcription factor pool in my nucleus? Do I have to worry about sort of competition effects, especially in bicoid, which has essentially thousands of binding sites and 10,000s of molecules at the early part of the embryo. And so because of that, I think it's like a little unclear, and I think it is actually valuable to find out what a single binding site can do. But yeah. Okay. So based on that, we then thought, you know, actually now what we've calculated is how much information does this binding site have about the signal, and how much information in this sort of information calculation can it carry about the cell fates. So now we have essentially both of these informations available that we would need to look at the information bottleneck. And I sort of said before, so sorry, yeah, the information bottleneck is the thing that I introduced before, where we now want to essentially maximize, sorry, and I see this should be PC given S because, yeah, but we essentially maximize the information of this binding site with respect to some output for a fixed sense of noise with which this binding site can measure the input. And maybe just to show this, we previously found, so one of the things that we've done previously was that we basically can infer the C for the gap genes, so for 4 input genes as opposed to for 1. And we can then look at how, you know, what are the features of these enhancers, of this compressed representations, and how does that compare to features of the enhancers? So concretely what we've done, so this is again the information bottleneck plot. So the x-axis is with how much noise do I measure? This is the thing that I want to maximize. Some regions of the plane are never accessible. And then here what we've done is we'd found this big black line from the data. We'd put in an additional constraint where we basically say, let's design binding site regions that can now only sense 1 transcription factor each. We'd optimally combine them, and we find that that combination is always worse than the one for, you know, where we just do the full compression of the 4 genes. So that suggests that this type of architecture would be suboptimal. And the biologists to some extent have said, okay, fair enough, that we kind of knew that, but I think it is nice that that came out from this approach as well. And so what we now wanted to see, though, this was entirely abstract. So we'd inferred C and we compared C to some abstract models. And now we wanted to see with this type of C, which admittedly is just 1 binding site region, where on this information bottleneck plane can I fall? So that's what I'll do now. And I'll start with this version of the Ising model, because here, basically, what you see is 2 things. So we've done the calculation for short times, like extremely short times, and for long times. And what you see here are different points for varying J. And you can see that for both, so for short times, again, you have like much less information, of course, because you're making a shorter measurement. For long times, you get much more information. You get almost up to the top. And what you see is that these curves are extremely close to the optimal bound of the bottleneck. And that should be a little bit surprising, but it is because I've partially not tell you something that Tamara had done, where she had essentially said, you know, let me not vary both J and EB at the same time. Let me actually do what the embryo is doing, and let me, for every J that every clustering strength of these transcription factors, let me fix the EB, the binding affinity here, such that the sort of steepest slope is in the middle of the embryo. Because she had said this is what we can really observe in the biology. Hunchback, of course, also has in nuclear cycle 13, so a little bit earlier than the gap genes that I showed, has like the step in the middle. And so she had automatically tuned EB accordingly, partially because she thought that was biologically reasonable. And I think that what's kind of interesting is now that just by this rational or sort of, well, yeah, rational biologically inspired choice, we can now see that, you know, tuning J with this constraint essentially tunes you along the information bottleneck line. And I think to me, that suggests maybe that this information bottleneck, or to some extent, these information optimizations are just like very intuitive ways to actually formalize mathematically, write down like something that just makes sense anyway. So this information bottleneck, in this case, you basically say that as you reclustering, sort of no clustering is here, as you increase the clustering, you tune along this optimal IB curve, and probably you want to be somewhere here where you basically sense with the weakest with the lowest possible information about your signal and still get the most information out. And that occurs for reclustering. And it's sort of interesting that I think the information bottleneck corresponds to some, or like can be seen as sort of moving the clustering string. So that is one physical interpretation. Of course, it's not the only one. But okay, so that was the work of the Ising model. And then with the other model, with the Hill function, we of course now can much more easily explore all sorts of parameters. So you can, pick random Ks, pick random Hs, pick random Ts, and throw them on the information plane. There again, you see that the short Ts give you less information than the long Ts in orange. And you see that random binding site constructs are definitely not going to be good enough. They're definitely going to end up somewhere in the information plane where you don't want to be. So the question was, now if we optimize, can we do better? And that again is true. So basically, if we find within our range and Hs and K the best possible ones, the one that optimize for this goal, you can see for the short measurement time, you're sort of very close to the info bottleneck region in this part. For the long measurement time, you're close to the optimal bound in this part. The reason why we are not achieving it here is sort of technical, and I maybe will skip that for now. I can explain it, but it's maybe not sort of super fun. It's essentially because these binding site devices, if you can get to this line here, but they are binding site devices that don't really make sense because they basically have occupations that never reach one. So you basically at some point you're saying, I can have a binding site that's just never occupied because my noise is actually so incredibly low because we've taken such a long measurement time that this guy is good enough. And that, we just thought, well, that doesn't really make sense. So it's a little bit, heuristic. But anyway, so what I wanted to say is you can achieve the optimal bound. And maybe I'll skip this part. And so overall, and then the final part that I wanted to say is, if you now look at a single point in this information bottleneck plane, like this point here labeled with 3, you can actually find that multiple binding site sensors correspond to that exact information. So same information about ICS, same information about ICX, even though these binding site sensors actually look pretty different. And I would say that again sort of suggests that often, multiple devices are optimal or similarly optimal. And maybe one can sort of interpret this a little bit in this direction of the stiff and sloppy modes. And then overall, I would say what we kind of hopefully show is that basically this reclustering, at least in this approximation, in this model, can help us achieve the information bottleneck bound. Okay, so then maybe to sort of wrap up this part, I guess what I'm hoping to have sort of told you is that the principle, to some extent, that we've used here is this information transmission. And we've said essentially 2 things, that basically it does sort of matter what information you're looking at in terms of, understanding whether a particular physical effect is optimal or not optimal. It does sort of really matter whether you're looking at the signal to noise ratio or some information about a variable. And I think that is maybe a little bit of it's great, but on the other hand, of course, if you optimize for the wrong thing, maybe that is, it will not give you the best possible intuition, and maybe that's a problem with the optimization approaches. But it's still sort of interesting to be aware of it, and I think interesting to know that maybe you can find a variable that makes sense, which I think in this case it does. Yes. 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 So I mean, it's maybe so basically you can see that the gap genes come up in nuclear cycle 12, so after 12 divisions, and they come up and they flicker, so it's super noisy, so you don't even see a pattern. And then in nuclear cycle 13, they come up, and at the end of nuclear cycle 13, they have a well-defined patterns. But again, at the beginning, it's a little bit noisy, but then it comes up. And then nuclear cycle 14, the last one, where I showed them as the input, that's where I would say maybe even, I don't know if there's been a lot of sort of detailed work on the protein level. There has been some work by Marianne Bauer from Thomas Gerger's group with data I've also used here to show a little bit how they come up, but she has much less embryo in these first, in the earlier time scales. So it looks more noisy, but it could also be more noisy maybe because there are less embryos available in that time scale. So I would say that the timing does matter. Like I think the longer, the longer time you live this system, the better it does, like sort of throughout development. And the reason why it matters here is sort of, I would say, it depends a little bit on how long you think this binding site has available to average, essentially. When you take occupation over a longer time scale, you can think of this as a well-averaged measurement of the transcription factor concentration. Whereas when you take like an instantaneous snapshot of binding, you basically maybe get by mistake that 5 of them have bound. If it now needs to sort of produce hunchback based on that, of course, it's going to be a noisy measurement. Is that? Mm-hmm. 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 Uh-huh. That's actually interesting. I don't know. That's actually very interesting. Yeah, I don't know exactly. Sort of also, yeah, nuclear cycle 14. But I don't know. Yeah, that's interesting. We should check, talk about it more, because I know you can change the temperature. I don't know exactly how that would affect it, but that's cool. Yeah. Mm-hmm. Yeah. Okay. Yes, exactly. And then the biology specific lessons, I would say, was that the timing here matters. And no, let me think, because now I wanted to talk about, there was an in-between part, in which I just actually, maybe I'll sort of semi-skip the in-between part because I wanted to have some time for the second part. So maybe the in-between part has sort of a summary here, where I guess what I wanted to say is that the hope with all of this is, of course, to predict to some extent enhancer features. And with this degeneracy at the optimum, I would say maybe we cannot actually find a single optimal enhancer construct rather than like a lot of different optimal enhancer constructs. But what we were hoping to do essentially, what I've shown you here is a lot of work with the models. What we were hoping to do also with Bill Bialek, with whom I have sort of a long-standing collaboration, also in my early postdoc work with, we thought it might be nice to sort of also try to do this top-down. Like, can we, look at the information bottleneck abstractly? Can we use that to infer certain enhancer features? And then maybe, can we do the same bottom-up? Can we look at sort of how in the models the noise maybe is tied to the model mean? And can we essentially maybe narrow down a little bit what set of crucial enhancer features need to be there? There's work that will hopefully come out later this year that I think is really exciting about multiple enhancers that I won't talk about. Just maybe to zoom you through this in like sort of 1 minute, because maybe it sort of is possible given that the introduction that I gave. So what we've done for a single binding site, so essentially this thing again, is that we were able to, we're basically saying, well, in a single binding site, I can make Gaussian approximations, and then I can actually write down this information bottleneck goal sort of semi-analytically. So in terms of, again, this entropy and the conditional entropy, where the conditional entropy depends on the noise, I can now calculate the noise because I've made these assumptions that things are Gaussian and monotonic and linear. And then I actually, I don't do this optimization that I've shown you this morning, not over PS, but simply about the noise, which in this case I can do because the noise is sort of constrained. And that gives me then, here, so this eta here also has this particular noise in Z hidden in it. And so then that gives me an expression actually for the noise, where the noise basically is depends to some extent on some function. I'm a little bit worried. Yeah, okay, fine. So in 1D, basically in 1D, it's basically a sum of the noise of S plus some map of S space into the output space. I don't know, you can look at the paper. But essentially what this allows you to do is that basically you then get essentially a master curve for the information bottleneck that just depends on 2 parameters, namely the information between the input and the output. And sorry, I'd use G here instead of S, and this parameter lambda. And you can see that, initially noisy information compressions will sort of collapse onto a single master curve if you basically rescale accordingly. And so now the hope is that since we were able to some extent to say, what you can do is to tune through this information bottleneck curve by only varying the noise. So in this case, we are not varying the mean, we are only varying the noise. We are wondering if we can apply this to now what happens if we would have a pair of 2 enhancers. And hopefully there'll be something soon. So anyway, so just to say that hopefully we can maybe sandwich these systems a little bit better into the model and the top-down inference phase. Okay, but with that, I really want to spend, or actually, are there questions, any more questions on the first part? Okay, So then what I would love to talk about is basically the second part, which my PhD student Olivier Ritterin has really sort of led. And it's a collaboration with collaborators at UCSB. And you can probably see that from the background. I keep thinking we should take a background of us in the rain, you know, in the Netherlands, maybe to just sort of bring out the contrast. But anyway, so they have like, they're very lucky to have a university by the beach. Anyway, so this is Max Wilson's lab, and they work on stem cells. And so I wanted to essentially tell you maybe how, or like the goal of the second work, which didn't fully work, but is maybe going in that direction, is how can we sort of optimize signals for stem cell populations. So the particular signaling pathway that I'm looking at is Wnt signals. Wnt is essentially a ligand that can bind to a cell receptor. We've seen a little bit already about these signaling pathways. So it's, I guess I just wanted to say it's a crucial cell differentiation regulator, and it regulates often with other additional factors. But in this case, the experiments have only Wnt as the single perturbative signal. So we're only looking at Wnt as an input. What our collaborators can do is that they have basically an optogenetic Wnt signal. So you can see that, you know, downstream targets of Wnt light up when the light is on. So basically, they optogenetically turn on this receptor. So that basically, instead of putting Wnt onto your cells, you just like optogenetically start this receptor. And that seems to work, that basically when the light is on, cells respond to Wnt. The problem is that the cells' response to Wnt is, you know, especially from my fly perspective, incredibly noisy. So, you know, basically as a function of time in hours, you can see that different cells do drastically different things in terms of output. And while I was initially sort of, you know, coming from the fly, I was initially not used to this, but it seems like, you know, even though this is here a synthetic construct, and so you might hope that the real system is actually a little bit less noisy, it's also culture cells and so on. But that these type of distributions are actually not uncommon in a lot of these gene regulatory systems. So this is, in this case, like a gamma distribution, super long-tailed, and so for us, a little bit worrying. And the reason how we got into this, which I won't talk about, is essentially our collaborators essentially found that basically as they do crazy things with these light pulses, like duration and frequency, they find some sort of intermediate suppression, and they asked us, why is there this suppression? And that part I will definitely skip because I don't have time. So we made some models, and it works because essentially if you put more parameters into your system, you can get the suppression if you tune the timescales right. But I think the question that I wanted to talk about here is to basically ask, you know, can we actually look at the synthetic population of cells and say, you know, what signals of Wnt can actually be responded to? And for me, the more useful question is, you know, can we actually do information approaches in some useful way if the noise is so high? Because I think that's really maybe to some extent a limitation that, like, you know, in the fly system, everything is super precise, but how does it work here? And these optimization approaches are sort of, I think, really in the spirit of this early work that Horace Barlow and others put forward in the sense of this efficient coding hypothesis that they had in the context of neuroscience, where they basically said that, you know, when you have a system that responds, the responses should be most sensitive to the most frequent stimuli. So what you see here on this plot is essentially data from, I think, Stephen Laughlin, who'd gone around and measured, you know, external images, like real external images, the contrast of these real external images on the x-axis. You can see that on top here. And then he said, like, how often, if I look at external images, how often do I observe a particular contrast? And, you know, there's, of course, a peak. Some natural images have a higher contrast than others. And then he'd measured the response of a particular neuron in the retina to these images. And you can see that that response curve is steepest where you have the most, you know, the highest density of natural images, which had sort of to them suggested that actually, you know, these things are optimized, they're efficiently coded. It does make sense for this neuron to see the most fine-grained where I also have the most responses. And we were sort of wondering, could we turn this around? Could we essentially just measure the input-output function of these noisy cells, and based on that, infer, you know, if these cells were optimized, what type of inputs they should have available. So for us, again, we now basically have a situation where we have an input, and it's not even clear whether this is the most biologically reasonable input, but fine. We have an input, which is basically the time duration of this Wnt signal. So how much Wnt do I have? We have an output, which is an expression of a single target gene, and that is this gamma distribution output. So you can see that it's very peaked around 0 for no Wnt input. When you have Wnt input on for long, it's like a very long-tailed distribution. And so the question was, can we optimize these signals so that with that noisy system, these cells can still have some precise wind to output map? So when we did that, we of course initially, so what you now do is what I did essentially in the morning, you do a prior optimization. So you optimize over the probability distribution of signals, which in this case is the probability distributions of time durations, and you say, what is the best distribution that I can use? So we've done a uniform distribution, and that of course is lousy. Like if you basically use all of these signals uniformly, you get like much less than a bit, which means that these cells cannot even tell, is wind present or not. Like it's completely impossible. But what is interesting was already when we basically said, like, what if we use two distinct signals? Like, what if we use essentially no wind and the longest possible wind signal that is reasonable given the lifetime of our cells in these experiments? In that case, you get more information. You get actually already 1 bit. So that, I think, again, you can understand maybe in the context of this noisy typewriter, like I have signals that are smeared all across. But basically, if I have sort of, the most differentiable signals, I can, of course, learn more. But so what's interesting is that we now, and this timing of these signals, that, where I get to close to 1 bit seems to be reasonable compared to also other experiments that basically when you have no signal, like either no wind or a 10-hour wind signal, that is enough for the cells to sort of differentiate. In our experiments, we haven't looked at this in detail, or we weren't able to look at, whether, or actually, I'll get to this, fine. So 10 hours was a reasonable time scale. But what I think is now sort of exciting is that Olivier then sat down and really sort of said, what is the actual optimal input distribution? For that, you use this Blahoud-Arimoto algorithm. There are some adjustments that he and also other people have made that I'll maybe get to. And then what you see is that actually, in this signal space, you actually should use 3 discrete signals where you have an intermediate discrete signals that you use at a lower frequency. So it's basically like in your typewriter, you now use A, Z, and maybe a little bit of L occasionally. So that seems to be optimal for this type of distribution. And so now our collaborators sort of suggested, maybe our measurements, maybe there's something else going on. Maybe these cells actually to differentiate have like a certain number of output genes that they then average over. Maybe these cells even average across neighbors. So, how would this look like if the noise was actually lower? For some mechanisms that could be biological, for some mechanisms that could be experimental. So we did that, and the trivial thing that you find, of course, is as you average, the information goes up. So this is you average over more cells or more outputs, the information goes up when you optimize the signal. So far, so good. But what I think was sort of exciting, because it also relates to this work that Ben Machter and others did in a completely different system, is that basically that you find this like really nice phase diagram essentially of prior signals, where as you lower the noise, you get more and more letters appearing, and it's sort of an interesting bifurcation of more and more letters, where in the limit of sort of infinite or very high averaging, you get this small noise limit appearing that I actually showed you on the board this morning. And in the work of Ben Machter, they had done essentially a paper where it's about data inference. So their paper was about, assume I am an external observer, I observe the outcome of a coin flip. So if, if I have a single experiment and my coin is 0 and 1, what should I estimate that this coin should be weighing? So in that case, if you get a 1, you should actually think this coin is always going to fall on head, because that's the single data point that you have. You shouldn't be thinking, oh, randomly this coin is like a 0.5 Bayes coin. And so they basically sort of interpreted this as basically when I have a few data points, I use the simplest most prior, which is, the strongest, most model-stated prior. And we were sort of interested to see if there's an analogy to some extent to our cells. So if our cells, do some amount of averaging, maybe, maybe at the very early beginning where you maybe only have almost 2 signals, in that case, what that would mean that if you're a cell and I get a signal that I can't really fully interpret, I should map it either on wind on or on wind off. And in our experiments, we couldn't check that, but there are experiments where these cells crawl around, and you can actually see that cells that experience wind form a cluster, which seems to suggest that they actually search for an environment that enhances their signal. And the other thing is that we are sort of assuming here that wind is essentially used for a binary cell-fate decision. So to some extent, you could say that that is a mini suggestion or so that these cells actually really try to interpret the wind signal in the most discrete or in the simplest possible prior model, similarly to how we should be doing inference. Yeah, and I think that was sort of for us the exciting part here, that we were wondering, you know, can we, I mean, maybe not exactly here where we don't know how these cells, you know, improve their averaging, but can we essentially find the best possible prior and then find evidence that that is also what the cells are doing? I can maybe quickly talk about these decoding maps because I find that also quite exciting. So one thing that when you do these information optimizations, what you could ask is basically, if I have a particular input signal, how well can I infer this particular input signal based on the thing that I know as the cell? So basically, in this example, it would be, how well can I infer the input signal based on how many transcription molecules are bound? Here, it would be, how well do I infer the input signal based on the gene regulation that I observe? And you see here that basically when you have a single cell and you make no assumption about the prior, so you assume a uniform prior distribution of signals, you have no chance. So like, you know, all a particular, if a cell has an input of 12 hours, there's not even, you know, there's not even a peak in the smear around 12 hours here. Whereas, of course, if these cells average more and more, you get a more precise peak around the correct input time. So the inference gets better and better. But the reason why I thought these decoding maps are useful here, or why Olivier calculated them here, was that you can actually see that as you do the signal optimization, you use the discrete priors. The slightly surprising thing is that you use more discrete priors than are uniquely distinguishable. And you can see this very nicely in the decoding map because there's always this smear, right? So if you had like in your typewriter, if you'd only used A and Z, and you basically said, okay, A and Z are completely indistinguishable, you know, this thing wouldn't have had any smear across. But what this optimization actually suggests is that you use slightly more symbols than you can uniquely distinguish and are okay with the smear. And Ben Marta and others have actually derived some sort of scaling law of this information with the number of priors that actually shows this, that you basically have more symbols than you can uniquely distinguish, which I kind of also think is interesting. In the context of potentially going in the direction of, can we interpret this in some sort of like modularity? Like, should we use more modules than we actually can uniquely differentiate? Okay, and the final slide is essentially, we had then looked at the sloppiness of the priors just because I felt like we had to. So we'd essentially wiggled these prior positions, or in the example of the noisy typewriter, you essentially maybe perturb A and B and Z and like whatever is next to Z, Y. And you sort of then calculate the information landscape. You see how the system responds to these priors, and you sort of similarly to before see that, you know, they span like a variety of different length scales, which means that some of these priors, you know, you can wiggle more, especially like when they are very weak, you can wiggle them, of course, a lot. This high one is this one that you can wiggle way more than the zero prior, and so you also get this sloppiness in the space of priors. Okay. So to wrap that up, essentially, I guess the part here would be to say that I think the physics lessons that we learned is that you can use these discrete signals to optimize information, and that you should use slightly more discrete signals than you think you might uniquely want to distinguish for optimal information flow. On the biology, I think what we learned is that actually these cells, even though they are noisy, if you pick the priors accordingly, you can get more than one bit of information out, which is enough for them to make these cell-fate decisions. And yeah, with that, I just wanted to end and sort of summarize. So here, yeah, I guess we've summarized before. And yeah, thank my funders and sort of say that I still think that I think maybe assuming to some extent optimality, even though we have to be careful with what we optimize and how, and maybe we should sort of really think about what optimization goals are consistent with each other and which aren't. I think that this optimality assumption can actually help us understand a little bit about the complexity in high-dimensional systems. And yeah, I'd like to thank both my group and then my collaborators whose work I talked about. Yes, and finally, sorry, if you haven't been to Delft, so I'm in Delft in the Netherlands, which is like 1 hour away from Amsterdam for people who don't know. And so the Dutch, of course, have this, you know, a lot of emphasis on the old painters. There's this particular painter, Vermeer, who painted this view on Delft. Delft has, of course, changed slightly since then, but I think one remark that I thought was interesting that I feel like I've learned in my time here. So the one thing that the Dutch painters are always sort of applauded to is this color scheme. People always say there are these unique colors, unique light shades, unique blues. And I think it is actually just because it's true. Like, you know, when you're that far in the north, this is what the light looks like. Like I was very surprised by this. I always thought they, you know, had some, you know, yeah, they were just good at painting, I think, the landscape. Anyway, okay, yeah, thank you very much. Are there questions? Yes. So yeah, so basically in the space of priors, we basically, so you have your signals, and then you have a probability distributions over these signals, right? And so in this case, because the priors are discrete, you can think of them as almost like delta function like things, you know, like here. And then what we did is that we essentially varied the position of this delta function, and or Olivier varied this position of this delta function and the other one. And you can actually, it's very nice, you can actually even write down the information, you know, as a function of these prior positions. So then this is 2t stars. You can do that actually analytically, and then you can sort of see how sloppy is this. So for example, this one, the one at 0, is actually needs to be at 0. This one here can vary a little bit more. And then as you add additional priors, you can of course do the same for all pairs and sort of ask how precise do I need to be here to sort of not vary my optimized information. Yes. So sorry, I basically then, yeah, so the typewriter is of course discrete. So I would say, you know, I basically just write my alphabet here in the signal space of different signals, so different signal durations. And then I would have said, you know, similarly to here, I use short duration and long duration. In my typewriter example, I use essentially A and Z. Yes, some continuous way. Yeah. Okay.